Hello, everyone. So thank you very much for inviting me today. So I'm going to talk about the large-scale neuroimaging studies and try to understand what are the interests and the limits of those large-scale neuroimaging studies. So first, I'll explain how we went from small sample studies to large-scale studies in neuroimaging. Then I think I'll discuss a couple of methodological roadblocks that I think are hampering us to unlock the full potential of those large-scale neuroimaging studies. Then I'll discuss if we need really big data or maybe more thick data. And then I'll discuss about what could be the future of those large-scale neuroimaging cohorts. And we'll be also starting one in the lab. So I'll just present it to you. So I'm mostly interested in the brain structure that is the cerebellum. I'm not going to talk too much about the cerebellum today, but I'm going to take this as an example to walk you through how we went from small sample study to large sample studies. So the cerebellum is, I think, a very interesting structure that contains more than 50% of the neurons of the brain. And that is involved in a broad range of cognitive function. Jeremy Schmaman, which is a South African neurologist, has discovered that patients that were having lesions in the posterior cerebellum here, as you can see with the blue dot, so either tumor or stroke, could have a broad range of cognitive and affective symptoms. And some of those symptoms are also symptoms of psychiatric disorders. It's also possible with cerebellar stimulation, at least in animal models, to induce or rescue ASD symptoms. Fact that I think is quite interesting is that if you're having a cerebellar injury at birth, it really increases the risk that someone will later develop autism. So we had many reasons to get interested to the structure of the cerebellum across neurodevelopmental and psychiatric disorders. And so we started by doing, I think at the beginning of my PhD, small set studies with typically something like 30 subjects. And then years after years, we saw the sample size increasing, mostly because we started to get access to those large-scale neuroimaging studies. And I think that's something that we have also realized while starting to work on small samples and working then afterwards on larger samples, is that in small samples, there is a huge, it might be really an inflation of effect size in small cohorts. And so basically, since 1993, there are more than 40 papers published that are asking exactly the same question, which is to know whether the cerebellar anatomy is different in individuals with autism and controls. And after more than 40 studies published, the meta-analyses are negative. The results are sometimes marginally positive. Sometimes it's also going completely in the opposite direction. What we found actually working in the EU AIM study, which is a large study of individuals with autism, is that when you taking the average in general, you're not seeing any difference, but it's possible that it's in some sites of inclusion, you're seeing, marginally significant effects, but I think that those effects are very, very small and they're not really reproducible. If you're changing the parcellation method that you're using, the results are not here anymore. So I think that what we've learned from large-scale neuroimaging studies is that when you're just studying a small sample, there's a really high risk, I would say, of false positive findings, mostly because we're dealing with a normal brain where a neuroradiologist is not able to make the diagnosis just by visual inspection. So we're really looking for small effects. And I think if you're just studying small samples, you might have false positive findings. So now we're getting access to a very large neuroimaging cohorts, such as the ABCD cohort or the Healthy Brain Network, which is a cohort that we've been studying a lot, that was launched by the Child Mind Institute in New York. And I think especially in neurodevelopmental disorders, what is interesting with those cohorts is that you can also do some dimensional or transdiagnostic analysis. Before, we were mostly comparing patients and controls. But for instance, if you're taking patients with schizophrenia, some of them are having alcohol abuse, taking cannabis. Some of them are having ADHD. There are many different diagnoses. A clear schizophrenia doesn't really exist. So I think that if you're just doing case control comparison, you might miss that. And for that reason, I think it's also interesting to have access to large transdiagnostic samples. And so in this healthy brain network cohort, we've been studying how the cerebral anatomy was linked to social abilities from a transdiagnostic perspective. And we found results that were actually surprising, meaning that the cerebral structure and function was mostly linked to general cognitive function and especially cognitive flexibility, much more than to social abilities or psychopathological traits in general. And so I think that this was interesting because it's really because we got access to this type of cohort that we were able to make this conclusion, which has been more difficult to make in small case sample studies. In general, I think that the results of large-scale neuroimaging studies are quite deceiving. There's this paper published by Mike in Nature showing that basically you need thousands of subjects to found a small association between brain and behavior, basically. So I don't think that with those small effect size, you're going to be able to have really meaningful clinical translation. I think also the question is, what do we want to obtain from those large-scale neuroimaging studies? Are we trying to find tools to predict the diagnosis? Are we trying to do prognosis prediction or to predict the treatment response? So that's, I think, an open question. It depends on every group. But so far, I think that the results I would say, are quite deceiving because we're only able to find association with a small effect size mostly. So I think there are also methodological roadblocks that are really hampering us to find more significant results, and I will discuss a couple of them. The first one is quality control, which is a very boring question, but I think also very important. So in our lab, we've been doing a lot of quality assessments. If you're interested in a single brain structure, for instance, the prefrontal cortex or the cerebellum, then it might be possible to do a visual inspection, to do something really careful to be sure that you're not missing anything. But if you're interested to the whole brain, it's really very, very time consuming, and it's hard to do a quality assessment on large-scale neuroimaging cohorts. So sometimes people tend to think that, okay, it's hard to do the QC, so you can just analyze the data, and because you have a large sample, then maybe you're going to find some positive results. But I really think, and that's what's been showing, I think, is that a careful quality check is having a huge impact on results, even if you're having a very large sample size. Another problem also, I think, is the reliability of neuroimaging metrics in general. There's a lot of work that was published showing that for resting state, for instance, you need to have a very long duration of scan to get your reliable measure. And most of the large-scale neuroimaging studies that have been presented have a very short scan of resting state mostly. So it might be also possible that if we're not finding very significant results, it might be also because the measure in itself is not very precise. And we've been trying to use tools to assess this reliability. So this is a toolbox developed by Qing Xu in New York. And basically you can map the ICC, so which is the measure of the reliability, but also the within subject variation and the between subject variation. The problem is that if you're studying brain features and you're dealing with brain feature with a very low between subject variation, it's going to be very difficult to do some meaningful brain association. If you're having a very large within subject variation, also it's the same problem. So I think that it might be really important to consider this when you're doing neuroimaging studies, and also when you're deciding of what will be the acquisition in this kind of study. So we've been comparing, for instance, cerebral partialation using this tool, and we were able to see that so those are the different lobules of the cerebellum, and we were able to see that for some, the measure was really not reliable. So we did this work using a retest data set. But so in general, I think that reliability is a very important topic for all studies in general, but especially when you're dealing with large-scale neuroimaging studies, because it's a huge financial investment. Also, I think a question is whether to know if we need big data. So what I mean by big data is like a lot of scanning acquisition, or if we need thick data. I think a major problem that we are facing is that mental disorders are really difficult to capture. And if you're just doing a clinical assessment with 1 or 2 scale one time, you're not going to capture an information that might be really meaningful. So for instance, using ecological momentary assessments might be an option to have a more precise clinical measure. And I think this also might be very interesting for large-scale neuroimaging studies. The question also is to know whether you need a very large sample size or you need a good reliability. And so this work, which is you can get access to the preprint, is basically trying to compare and to assess this. So it's run mostly on the ABCD studies and also on additional simulations. And so basically, I think the interesting message of the paper is that if you're having a small sample to maximize the effect of brain and behavior association, you need to increase the sample. But after a certain sample size, then it's better to maximize the reliability of both the clinical and the neuroimaging measures. In a way, we don't necessarily need, I think, thousands and thousands of subjects. We also need a very good measure on both the neuroimaging and the clinical part. And this is also, I think, an analysis that I think was quite interesting. It was the idea was that if you're from a funder perspective, if you're investing money in a large-scale study, do you need 100 individuals? Do you need 100,000 individuals? Or do you need 1,000 individuals with 2 clinical assessments? And so what this figure I think is nicely showing is that if you have 2 clinical assessments, or if you are increasing the quality of the clinical assessments, then it might be more impactful than just scanning more and more and more subjects. So what then could be the future of large-scale neuroimaging cohorts? Well, I think that first we'll get a very large funding in France, which is named the PEPR PROPSI, that is led by Marion Leboyer. And basically the idea is to implement precision medicine in psychiatry. So the goal is to develop new diagnostic tools, new therapeutic strategies, and it would be heavily relying on a big cohort, which is named the French Minds cohort. That is, I think, going to be helpful to try to build new diagnostic tools and therapeutic strategies based on this large cohort. And so in this context, we started thinking with a group on what should be in this cohort. And I think it's a very difficult question, because the problem that we're facing is that we're deciding of a protocol now, then the data are going to be acquired in 5 years, and then only we're going to start to analyze the data. So we really need to make sure that the choices that we're doing today will be the good ones, because we don't want to regret any choice that we could do. So we'll be relying on the CATSI neuroimaging platform for harmonization and to calculate the reliability. What we're trying to do also is to push forward the methods that we're using. So we would like to use an fMRI naturalistic new viewing paradigm, also coupled with a proxy for a measure of eye tracking. The idea is that when you're doing a resting state, you're just studying the brain at rest. It's like when you're doing an EKG just at rest, it might be good to have some information, but you're not really seeing how the brain or the heart is functioning. So I think in cardiology, for instance, you can do a stress test where you'll be running on the treadmill and see how the heart is behaving with the effort. And I think it's kind of the same idea with naturalistic viewing paradigm. It's that you need to be able to measure different brain states depending on what you're seeing and doing maybe different types of analysis than what you could just do with resting state analysis. We're also really interested by ultra high field neuroimaging because it would also enable us to visualize certain brain structure that are difficult to visualize with a 3D MRI. And for instance, deep cerebral nuclei are very interesting structure if you're trying to understand cerebral networks, in general. And so you can now with 7T, you can really visualize those deep brain structure and I think better understand in general the functional connectivity. And also what we're really interested in is to use normative models. So the idea is that in those large cohorts, we might be able to build normative models that would enable us to identify certain subjects that will be deviating from the norm, and that we could maybe propose to certain subjects some specific targeted interventions. So having large-scale neuroimaging cohorts is also enabling you to have access to those normative models and to try to implement some precision medicine approaches. So to conclude, I think that I don't think actually that large-scale neuroimaging cohorts are going to be able to provide ready-to-use biomarkers for neurodevelopmental and psychiatric disorders. I think it's only a tool, but I think in itself, it's not going to have like a direct clinical translation. But I think that those are useful to understand the association between brain and behavior and also their determinants. And maybe also to try with some approaches such as normative modeling to transfer this information to smaller data sets when you're trying to implement a therapeutic strategy. I think there are still many challenges to be overcome, such as harmonization, also finding a good way to do a meaningful quality assessment, and also to try to include more reliable neuroimaging and clinical assessments. And so I really think that we still need some small studies to test more specific hypotheses, because when you're building a large-scale neuroimaging studies, it needs to be used by different type of researchers that want to ask different type of questions. And also, I think we still need small studies to, for instance, study the effects of a therapeutic intervention. So thank you very much for your attention. And I would like to thank Jocelyn Ouenou, Marion Leboyer, Mike Pira, my supervisors, and also the students that contributed to this work. Thank you very much.